My name is Ryan Sarkisic, and the title of my project is Global Warming Mitigation, Optimizing CO2 Removal of Algae Farming. Global warming is one of the biggest problems that we currently face and is usually caused by an excess amount of CO2 in the atmosphere. One of the main ways that nature is able to combat CO2 in the atmosphere is through photosynthesis. Algae, more than any other photosynthetic organism, is responsible for taking CO2 out of the atmosphere. And in recent years, there's been a lot of research to try to find the optimal ways to grow algae, including the pH, light, and temperature of growing conditions. However, there was nothing I could find about the optimal light-dark cycle, because the general consensus is that it's 12 hours on, 12 hours off. So the question is, what is the optimal light-dark cycle of Nannochloropsis, the algae I'm going to use? So the hypothesis is, as I stated, the general consensus of 12 hours on, 12 hours off. Each 16-ounce mason jar had the same amount of nutrients, salts, and Nannochloropsis culture and water. Each of these jars was able to have a different duty cycle and period. And the jar on the left is the one with continuous light exposure. The jar on the right did not have any light exposure, and the jars in the middle all have different light cycle times and exposures or periods. The first graph shows the amount of growth for the main four. The one that's always on, the one that's on for 16 hours and off for 8 hours, the one that's on for 12 hours and the one that's off for 12 hours, and the one that's always off, which is the control. The one that's on for 12 hours and off for 12 hours is performing considerably worse than when the light is always on or when it's on for 16 hours and off for 8 hours, showing the need for continuous light of Nannochloropsis algae. The second graph shows the average amount of algae growth per day in the first week. This reflects what's seen in the first graph because you can see when the light is only on for 12 hours on and 12 hours off, you see that it grows worse than all the other ones except for the control. As you can see, the one that's always on had the highest growth rate, and the one that was on for 16 hours had the second highest. The third graph represents all of the different graphs that had the same duty cycle, which means that they're technically on for the same amount of time. However, they have different continuous light exposure. And here you can see that it actually matters the more continuous light exposure that you have, because the longer it was on continuously, 16 hours, it actually performed slightly better than when it was on for 8 hours and off for 4 hours, or on for 1 hour and off for 0.5 hours. The fourth graph shows the algae growth life cycle, which consists of the lag phase, the exponential phase, the stationary phase, and the death phase. Usually, at the beginning of the stationary phase is where you harvest your algae. And if you see here, you can see that on day 9 is when you harvest for when it's always on, but for when it's on for 12 hours and off for 12 hours, you see that it's only harvestable until day 12 and at a lower density, meaning that there's less to harvest. And when it's on for 12 hours and off for 12 hours, it only reaches full maturity at day 17. So, what's to learn from this experiment? Well, basically, the more continuous light exposure and the higher duty cycle is given to Nannochloropsis algae, which is single cell, the more it grows, meaning that it doesn't need a dark cycle. So what's next? Well, as I said before, different light spectrums have been tested on with algae, and it's been found that green algae, since they reflect the green light, basically green light is rendered useless to them. So using a blue-red light could drastically influence this experiment's results. Also, finding the light saturation intensity, and the good saturation intensity is 120 micromole per meter square, but finding a specific one is really good. And what a light saturation intensity is, it's basically how much light can the algae have before it has to work to get rid of it. Also, studying the effect of high-frequency light cycling, which is turning on and off the light very fast to see if that influences the growth of algae at all, and repeating the experiment with different types of algae, such as spirulina. Because Nannochloropsis is only singular and is very strong, known as a bowl of algae, it would be interesting to know how different algae responds. Thank you.